to have Aurelien Roux from University of Geneva. Aurelien is really a world leader in applying biology and physics to study the mechanics of membrane trafficking, with a special focus on dynamine, clathrin, ESCRT-III, which we'll hear about today, and also as of recently in the mechanics of growing epithelium. So just a little bit about Aurel. He did his PhD in Paris at Institut Paris. Hi, somebody's coming. Okay. And then he did a postdoc at Yale, after which he started his CNRS position in Paris, and then moved to Geneva in 2010, and he's been there since. And today we're very much excited to hear about the details of this beautiful movie. So yes, Aurel, please go ahead. And I'll just say that if you have any burning questions, please feel free to ask, raise your hand, or maybe even just speak up if nobody's paying attention to you. But otherwise, we'll wait until the end and I'll read the questions from the chat, or you can just speak when called. Okay, Aurel, please.
Thank you, Wendela, for this very nice introduction. Yeah, it's a pleasure to be virtually with you. It's not nice not to be in real with you, I have to say, but that's how it goes with the current pandemics. I'm still very glad and happy to share all this work with you today, and at least you, I hope, will be able to see it again and again and again on the TV, since we at least can record that. So feel free to interrupt. Yes, again, as Wendela said, just raise your hand or interrupt me vocally, because I don't see you. So please feel free to interrupt. The escorts, the four complexes of the escorts have actually been found during a screen that was conducted in the lab of Scott Emmer about 10 to 15 years ago now, no, 20 to 25 years ago. And it's all about the formation of these tiny vesicles that you see here in the luminal part of the endosomes. And I hope you can see my pointer. This is one of the intermediate in the endosome to lysosome pathway, the multivesicular bodies. And as you can see, they're filled up with tiny structures which are called ILVs for intraluminal vesicles. And these ILVs actually contain all the material that will be degraded when the multivesicular bodies fuse with lysosomes, in order to say these are membrane component carriers for recycling purposes. And they are formed, as you can see from this nice picture, by budding from the outer membrane inward to form these vesicles. So functionally speaking, the lumen of these ILVs is topologically speaking connected to the cytoplasm, and of course all the proteins that will trigger the formation of these ILVs have to come inside the bud, and that's what this drawing is supposed to show. And the proteins that have been found during the screen by Scott Emmer to participate in the two steps of the formation of these ILVs, the deformation and the fission, are actually belonging to the ESCRT-III complex. ESCRT stands for endosomal sorting complex involved in retrograde transport number 3. Now, this ESCRT-III turned out to be extremely well distributed in membrane function in the cell. It's actually everywhere. If I cite now James Hurley about this, it turned out to be essentially involved in all the fission reaction that involve proteins found in the neck, inside the neck of the membrane that needs to be cleaved, which is the opposite orientation of the dynamine, if you wish. And in particular, and I'm going to show only 2 examples, which are probably the best described so far, besides the ILV. The first is the virion. So the virions will bud from the membrane, the plasma membrane of the cell, and actually the gag envelope will, in particular of the HIV, will hijack the ESCRT-III complex to cut the neck that connects the virion to the cell, and that will ensure that the the virion is liberated from the cell and will be able to infect a new cell. And the second reaction is actually a crucial step in cell division. It's actually the last step of cell division, the one that breaks the tiny bridge of membrane that connects the two daughter cells. Abscission, it's called cytokinetic abscission. And this is also mediated by the ESCRT, and you can see beautiful pictures from Daniel Gerlich's lab, where you see one component of the ESCRT-III complex forming this conical structure, and the fission or the abscission will occur exactly at the tip of this conical structure. This is a tomographic representation of the complex, which shows that it's organized as conical or helical filaments in the neck. So the conclusion of these cellular studies is that ESCRT-III is a membrane remodeling complex, and it's essentially the only one that is able to work on every membrane: the nuclear membrane, the plasma membrane, the endosomal membrane, and most probably also the ER or the Golgi membrane to some extent. So that's very unique to what is known about membrane remodeling complexes. If you compare to clathrin, which works only at the TGN and at the plasma membrane, if you compare to dynamine, which is essentially bound to plasma membrane, or the COP1 and COP2, which are Golgi and ER based only, the ESCRT is really unique in that regard. It's also unique, and that's the purpose of this slide, because it's the only membrane remodeling complex present in archaea, and archaea are proposed to be organisms that are in between prokaryotes and eukaryotes. At least the Loki archaeota is proposed to be the most ancient eukaryote found so far, and it belongs to a transition in the living tree between the eukaryotes and the other archaeota. And what is very impressive is that the only membrane remodeling complex present in most of the archaea is actually the ESCRT-III. Okay, so that argues, and actually one of your colleagues, Bursbaum, has been proposing that the ESCRTs could be involved in the transition from prokaryotes to eukaryotes by forming the endomembrane system through a reversed symbiotic reaction that is presented in that very nice paper that I strongly encourage you to read. And this is very hypothetical for the moment, but it brings a lot of perspective on how we think about the structure of cells. So the ESCRT are very unique because they're very ancient, and very unique because they essentially work at any membrane. So of course we're interested in understanding how this works. And I'll present very briefly the structure of the complex in terms of subunits, and I'll try to simplify it because it's actually very different from one organism to another, and it's also not very well understood how you can classify those proteins by subgroups. So that's my interpretation, and it's also in the purpose of simplifying the discussion with you, but keep in mind that this is maybe a bit of a caricatural view of the structure. So there's a first layer in the complex, I would call it layer 1, that is composed of nucleators of the complex, the VPS20 for yeast people, and SNF7, which is one of the main components of the polymer. There's a layer 2 that is composed of two subunits that work together to form a subcomplex, and this is pretty well established, VPS2 and VPS24. And there's a third layer that is composed of at least two components, D2 and IST1, which are presented here. Again, this is the yeast component, so you can see that there's like 6 proteins, but in mammals it would go up to 8, and you have archaea that have down to 2 components only. But they all work with one big ATPase, which is the VPS4, and this VPS4 ATPase, sorry, is involved in making a turnover or dynamic instability in the complex, a little bit like what is seen in actin or microtubules. One of the goals of this, what I'm going to show you today, is to understand what's the exact function of this turnover and what does it bring in terms of mechanism. This is work we did with Daniel Gerlich a few years ago. So we started working on the ESCRT-III quite some time ago in the early 2010, and that's when we saw this beautiful picture from Phil Hempston's lab and John Heuser's with John Heuser's technique, where they transfected cells with CHIM4B, which is actually the mammalian equivalent of SNF7, and they observed these beautiful spirals at the surface of the plasma membrane, the inner leaflet of the plasma membrane. You can see clathrin here, you see those nice spirals here. And what was even more striking is that when they overexpressed the SNF7 and blocked the VPS4 function, meaning that the SNF7 would massively polymerize at the surface, then they see membrane deformation. They saw membrane deformation. What they observed was these protrusions. So here you're looking at the external leaflet of the cell, and you see protrusions coming towards you. I guess you understand that the color code calls for a 3D view of the image, which is not available today, unfortunately. And then what they did was to wash out all the lipid using detergent, and they saw that the protrusions were actually coming from the central part of the spirals as a tubular structure that was protruding from the central part. And so this came with the idea, and that was an idea we had with Martin quite some time ago. Martin is our long-term collaborator, and he's a physicist, a theoretician. And so we imagined that these structures could be formed by buckling of elastic spirals. And I'm going to explain this with this slide here and show you a little movie about it later. So the idea is very simple. If you have a polymer that grows with a certain preferred radius of curvature, but as so this is one of the essential feature of this filament. It's a curved filament, and it has some elasticity. Then it will continue growing on the flat surface as a spiral. And doing so, it will accumulate elastic stress because the inner filaments will be more compressed and the outer filament will be more stretched. And one way to release the stress that accumulates through growth is actually by bulging the membrane, buckling the membrane, and forming a tubular structure where most of the filaments, which are shown in white here, are actually relaxed. Only the very top ones or the very bottom ones are a little bit straight. So what Martin did was to put this into equation, as you can see, as a competition between the elastic energy of the filament in regard of the elastic energy of the membrane, and could compute and design the shape diagram of this. So we wanted to test this hypothesis, and the way Nico did that in the lab was to grow patches of membrane, patches of protein at the top of supported bilayer. The bilayers are shown in gray here, or white gray, and the proteins are shown in green. And what he observed is that circular patches of SNF7 could grow at the surface of these membrane, and you could follow the growth by using chymograph structures. And those patches are actually filled with tightly packed spirals of filaments, as you can see from this AFM image. And we could even track the growth of these spirals live using high-speed AFM, and that was done with Simon Schering, and was a highlight of this paper. And we could show that the growth dynamics, but also the relaxing properties of those spirals is actually compatible with those spirals being spiral springs. So how does this work? We think the way it works is pretty simple. You have a main protein SNF7. It forms a curved filament at the surface of the plasma membrane, And by growing, it actually forms spirals that accumulate stress because the inner filament are overbent and the outer filament are actually stretched. And to release the stress within the spiral, you would have buckling, forming this invagination or protrusions that look like tubules. And of course, these structures are filled with the filaments. Now, there was one problem still. The problem was that we never observed cylindrical membrane structures with SNF7 alone, meaning that we did not observe membrane deformation, or I would say large deformation with SNF7 alone. But we had a hint from a study from Scott Emmer, and this is the paper where the data are showing it, that actually deformation could actually be triggered by the addition of other subunits in the complex. And that's what you see here. You see filaments of SNF7 alone, but then when Mike Heaney and Scott added the second layer of the complex VPS24, VPS2, they observed the formation of these helical filaments here at the surface of the membrane. So this is supposed, and that's what they say in the paper, and I've talked with them quite a bit about it. They say this is without membrane inside. Maybe there's still membrane inside, but I'll show you structures where there's definitely membrane inside, and that looks very much like this. So again, we were trying to understand what was going on here. And as I said, with SNF7 or layer 1, we had only a flat spiral. And this is a nice picture from Nico's work where you see a crushed liposome on the surface, and you see that the spirals are essentially flat at the surface of the membrane. And these spirals, SNF7 spirals, are actually made of single stranded filaments that spiral into these structures and can form locally double-stranded filaments, as shown in yellow. Now, when Nico and Joachim, another postdoc in the lab, added VPS2 and 24, they observed the formation of a second filament along the pre-existing SNF7 filament. So essentially, what we're saying here is that VPS2 and 24 is forming a secondary filament along the previously existing SNF7 filament. And that gave a little bit of some ideas about how deformation could be triggered by addition of VPS2 and 24. And that was actually the work that Joachim undertook in the lab. So the idea is pretty simple, is if you add layer 2 to a growing spiral of layer 1, maybe you change either the architecture of the entire spiral by, for example, giving an additional twist that will transform the spiral into a helix, or you could change the mechanical properties of the helix. For example, you could increase its rigidity so that it would be more prone to buckling. So that was the initial hypothesis we had. So what Joachim did was to actually incubate for a long time layer 1 and layer 2, meaning SNF7, VPS2, and VPS24 together with liposomes, and he observed these amazing spiraling cords that you see here that look very much like ancient phones that still had wires for the ones that have lived in another time. And those wires actually, when he looked at them through cryo-EM tomography, clearly presented a harness of filaments that were spiraling around the membrane tube shown in green here, as you can see. And this is a membrane, I would say a hand or observer segmented image, and this is just an averaging structure to really show that the filaments are visible without any particular treatment or subjective selection. So what did we learn about this structure? Well, first we could get a fairly good view on how the filaments are organized onto the spiraling tubes. And I hope you can see from these class averages that we have essentially two groups of filaments that are actually forming double-stranded filaments. We have the equatorial group in blue also shown here, and the red group is the polar group. And as you can see, they are not oriented exactly the same onto the membrane because, of course, the membrane tube is circular. And that, if you now look at the structure in a more conceptual way, since all the filaments are following the same helical path, that actually means that if, with the assumption that all filaments are equivalent in terms of subunits and composition, that the filaments that are equatorial will bind the membrane with the blue interface, whereas the polar filament will bind the membrane with the orange interface. And that's very intriguing because that suggests that the single filaments of proteins would actually be able to bind the membrane with different orientations, and that they don't have a single interface with membrane. So can we see that in samples? Actually, Joachim added detergent to the soup, and he observed spiraling filaments that at first sight were not filled with membrane, as you can see from this. And he could see this very nice zigzag type of filaments, a more sinusoidal form, and he could see also large extent that were more looking like ribbons. So it turns out that this form is actually the polar form of the filament. This one is actually the equatorial form, and we could not get a high resolution of this structure, but we did get with Adam Frost at UCSF a high resolution of the polar form. That's what I'm going to show you now. And what you can actually see is that the structure contains membrane. You see the gray white part here is actually a micelle of membrane that is stabilized by the high concentration of detergent, and it's sandwiched between two filaments that have the polar structure, essentially, because they're spiraling exactly like the polar filaments that we see in the tubes. And what you can see, I hope, is that the orientation of the subunits is very easy to see on those on this structure. So it's essentially binding with this certain angle. These are single subunits taken from crystal structures that have been opposed just to show you that they look very much like what we see in the structure. And to give you a better understanding of what that structure means, it essentially means that the subunits are binding with a 90-degree angle compared to what had been published before by Adam Frost. And there's actually a new paper on this with the membrane inside, but you can see that it was proposed that these proteins will bind laterally. So the blue part is the positively charged part of the protein that binds to negatively charged lipids. And so the binding phase on this structure is actually through a tube that would go into the helical structure. And that's 90 degrees of the binding of this filament, where the membrane is actually oriented at 90 degrees compared to the same filament. So that's very interesting because essentially the structure tells you that the filaments are already able to change their orientation towards the membrane, which is completely uncommon. Now, what we did with Martin was to look for stability, mechanical stability parameters of the helical tube. And this is not expected from a purely mechanic point of view, because you expect that if you have a spiraling filament, you would actually form a straight tube inside this spiraling filament, as it has been shown before for escorts, but also for dynamine and for other bar proteins, for example. So it's expected to have a straight filament, a straight tube inside a helical filament, not a spiraling or helical tube inside a helical filament. It turns out that Luca did a great work on this theoretically, and he could show that for high enough tension, the spiraling form becomes more stable than the straight tube form, essentially because you will have less cost in terms of curvature in those structures, as surprising as it can be. What it did was to even calculate the change of parameters that we would expect from the pressure of the membrane inside a structure made this time with 3 filaments, 1 equatorial and 2 polar. And he could show that the dimensions we measure inside the sample in vitro, so by cryo-EM, accounts for the fact that there is 2 essential parameters that are changing. And these 2 parameters are part of the 3 ingredients, of course, an helical scaffold, but that the bending rigidity and the twisting rigidity of the blue filament is different from the red filament. So essentially the equatorial filament bend with a different rigidity than the red ones. The polar filament. And the second finding, which I think is very interesting, is also that the binding energy to the membrane must be different between these 2 filaments, again agreeing on the fact that these filaments bind the membrane with a different interface. So that was confirming somehow the findings that we had thought of seeing the structure. Okay, so what I've told you now is that addition of VPS2 and 24 rigidifies the SNF7 spirals. It could actually give also an additional twist, and I'm going to come back to this in the later points that I will show. And what it means, giving an additional twist, is essentially that a flat spiral with a minimal twist in the Z direction would actually increase its twist in the Z direction so that it buckles. The filaments can also bind the membrane in at least 2 orientations. Actually, if you look all the data available now from crystal structure from Scott Emmer, cryo-EM structure from Adam's Frost lab, and our structure in collaboration with Adam, we can see at least 3 orientations. So it's even more than 2. So we thought that buckling could be triggered by increase of rigidity and increase of twist upon VPS2 and VPS24 binding. But there's the problem of the dynamics. As I told you, one important and essential player for fission is actually the presence and the activity of VPS4. And so we were wondering how can we couple these mechanical at equilibrium finding about the structure of the filament with the dynamics induced by the VPS4 molecule. And so one of the thing we thought was that the sequential copolymerization of subunits with different equilibrium state, one forming, for example, flat large spirals and the other one forming tight helical structures, could actually lead to the formation of these conical structure that are observed in vivo and at least also in some example in vitro. So Anna, in the work I'm going to show you now, is mostly her work from PhD, and she just got the Bernstein International Award, which is one of the most prestigious international award in Europe for PhD work. So what Anna did was to use the assay developed by Nicola, but then add on many more components of the ESCRT-III, including VPS4, to trigger the turnover of those subunits. So just to remind you, she was taking giant liposomes, explode them on supported bilayer, and then she could look at the growth of all these subunits at the surface of this supported bilayer. And here you see the example of forming patches with SNF7, and then added VPS2 and did 2 to see and look at the binding of those proteins at the surface of the SNF7 patches. You can look at the chymograph, and doing these experiments, she could show that actually VPS2, which was known from before, VPS2 and VPS24 have to bind together at only a membrane that is covered with SNF7. So essentially, SNF7 recruits the second layer of the complex VPS2 and 24. And then she could show that, again, D2 and VPS2 work in tandem to bind to VPS24 and VPS2. I won't go into the details, but that suggested that layer 2 is indeed recruiting layer 3, and one of the components of layer 3 IST1 is only recruited by DID2 once DID2 is bound to VPS2 and to the previous complex. So there's a sequence of recruitment that seem to occur in those simple without VPS4 experiment. Now, what was striking as well is that this sequence of recruitment was exactly the reverse when Anna added when Anna looked at the dynamics of binding. And it turns out that VPS2 and VPS24 were binding extremely fast to SNF7. DID2 and IST1, which are layer 3, took more time to bind to SNF7. So we then looked whether this sequence was kept during the depolymerization or the turnover principle. So what Anna did was to actually put all complex together, incubate them quite some time onto the supported bilayer so that they all assembled together. And then she added VPS4 plus ATP and looked at the depolymerization of the complex and how fast each subunit would come off the complex. And what she observed is that SNF7 and VPS24 were going out very fast. It took a little bit more time for VPS2, and DID2 and IST1, which is essentially layer 3, was staying very long time at the surface of the supported bilayer. So it seemed that you had a sequence of recruitment, and this sequence of recruitment was compelling with the sequence of depolymerization we would see. Essentially that the early comers to the membrane, layer 1 and layer 2, would be coming out of the membrane earlier than the later comers, essentially components of layer 3. So to show the sequence completely, what Anna did was to generate SNF7 patches and add all the other subunits, including VPS4, at the same time on these SNF7 patches. Now, she looked at the fluorescence of SNF7 in green as a marker, as a reference for the time. And she looked in different experiments at different subunits that were labeled. All this in a buffer containing all proteins. And what she observed was that VPS24 was essentially coming up very quickly after the addition of all proteins and coming off very quickly. VPS2 came after SNF7 started depolymerizing and took a little bit more time to go away. And again, DID2 and IST1, and I want to mention it that for this experiment, she had to take another reference, which is VPS2. She could see that these two proteins were coming later. So with this and renormalizing the time by the SNF7 reference, she could organize the entire sequence that is occurring at the same time on the surface of the bilayer. And what you can see here is that essentially when you start with from SNF7 patches, SNF7 and VPS24 coming very early, peaks very early, then VPS2, then DID2 and IST1 are coming a bit later. So the sequence is shown here. That's the organized sequence as a drawing or as a scheme. And I think that's one of the only example of sequential recruitment of a whole series of membrane remodeling components at the surface of membranes in vitro. So what I'm telling you here is that SNF7 is recruiting VPS24 and VPS2, and I want to point out the pivotal role of VPS2, which allows for the exchange of VPS24 with DID2, and then SNF7 moves away, and IST1 is then recruited by the DID2 VPS2 complex. It's only when DID2 and IST1 complex, and I'll come back to the role of this last complex at the end, it's only when it's formed that VPS2 can go away. That's the sequence. So a striking feature was that when she looked at the very last complex that I showed you, which starts with VPS2 and DID2 and ends with DID2 IST1, she could observe that those molecules would stay longer at the surface of membranes, whether it was supported or, in this example, on GUVs, on giant liposomes. It would stay longer, but not only it would stay longer, but it would also form clusters that seem to deform the membrane. I hope you can see the membrane staining here in white, and you can see that with time it forms clumps of membrane, which are actually a deformation or invagination of membrane. And this she could look at by EM using two techniques. One technique is standard negative stain, as you can see from the bottom images, and the other one is freeze fracture, as you can see from the you can see a negative stain from the top images and a freeze fracture from the bottom images. So the layer 1 plus layer 2 would form very shallow structures. And when you add components of the layer 3, or when you exchange components of layer 2 with components of layer 3, she could see increase of deformation of the membrane. And I hope you can see that when you look from the surface of the vesicle, you actually see by negative stain a dark spot in the central part of the spiral, and that's because there's an invagination going inwards. And if you now break open the liposomes and you look from inside the liposome, you actually see a protrusion towards you, meaning that the spirals that are binding outside the liposome protrude inward the vesicle. So what I'm telling you here is that essentially we form a layer 1, and when we add layer 2, we start buckling the membrane, and the buckling transforms into a deep invagination when layer 1 is exchanged with layer 3, and that's essentially dependent on VPS4 and ATP. Well, so what we knew at this stage is that the ATPase VPS4 triggers a sequence of recruitment and recycling of subunits. The sequence of recruitment is coupled to progressive membrane deformation. And we think, and I'm going to show you a few more evidence of this, that the addition of other subunits can rigidify ESCRT-III filaments, and also it could give an additional twist to the structure. So let's go back to the twist story, and let's see how this could help the formation. So we were impressed by the work of Andela that she published about a bit more than a year ago, where they modeled these nice flat spirals, and by just twisting the filament and the interface with which the filament binds to the membrane, she could observe the formation of a shallow and then deep invagination. So we wondered whether this mechanism could be the same than the one we observed, but we had a few difference in the way the twist would be transmitted. One way we thought could be actually the case occurring in our experiment would not be that you have a mechanical twist increasing with time in a pre-existing filament, but rather that you would have a first filament coming with a certain twist, and that you add a second filament with a different twist, and you remove then the first filament, letting the second filament to relax to its preferred shape. And indeed, that's what Andela and Sharon did in simulations that you see on this slide. And you start with a filament that is forming a flat spiral. And if you add a second filament which prefers to bind into a tubular shape, you initially find a shallow invagination when you bind the second filament to the first one. And if you remove the first filament, then you end up having this deep invagination. So we thought this could very well explain the experiment that Anna had seen by EM and sequential recruitment of proteins. Now, to show that there was actually a twist in the filament when we added the components of layer 2 and layer 3, what Joachim did was to actually look back at filaments, spiraling filaments with detergents when you had VPS2 and 24 and SNF7, or whether you had SNF7, VPS2 and DID2. And I hope you can see from the class averages that the filaments formed with DID2 are actually more twisted than the ones formed with VPS2 and 24 only. And that suggested that when you add components of layer 3, you indeed increase the twist. And also it shows that the twist of VPS2 24 SNF7 filament is higher than SNF7 alone, which is just a flat spiral. So we think that the twist is actually an important component of the buckling mechanism. So this nice video from Nicolas Dimitri was at UCL was actually explaining this dynamically. So if you form this first spiral containing the two types of filaments and you let go the initial filament, then you see a sharp transition of the membrane shape from a shallow invagination to a deeply curved invagination. And we think really that's the essence of buckling the membrane. So why do we need this last complex IST1 DID2? Well, we need this complex most probably to actually generate fission, and I hope I'll be able to convince you in the few last minutes we have. So what Anna looked at, and I hope you can see this is a broken open vesicle with tiny structures, which are actually tiny protrusions coming out. And you see some of them here. And these protrusions are actually made of the entire sequence adding VPS4 ATP. So that means that the last complex left on those membrane is actually DID2 IST1. And this is about 2 times more constricted than the VPS2 DID2 complex, which is the previous occurring subcomplex in the sequence. So it seems that adding IST1 generates constriction of the membrane. And indeed, that's known. And this is a paper published the same year by Adam Frost, where they analyzed by cryo-EM the structure of CHIM1B proteins, which are actually the mammalian equivalent of DID2, and CHIM1B IST1 copolymer on the membrane. And so this is CHIM1B on the top and CHIM1B plus IST1 on the bottom. And I'm sure you see that it's much more constricted when you add IST1, and the radius, the inner radius, is about twice smaller, which is exactly what we observed in our not cryo-EM, but negative stain images. What is interesting is that this structure actually, even though it's not yet at the same level of constriction than the dynamine one, but at least gets much closer to a fission state, which is this highly constricted state that is required for fission. And so we think that that IST1 and D2 forms the fission competent subcomplex. Is that the case? Well, Anna did a very simple standard assay to ask the question whether this sequence leading to the DID2 IST1 complex would be there to make the complex fission competent. So what she did was first to use the pre-last complex in the sequence, the VPS2 DID2, to form tubules on liposomes. And these tubules, I hope you see, form sorts of constricted or pearled tubes with constricted nape, where we can see a tiny complex on top of this. And when she added IST1, she didn't see much of a change, but when she added IST1 plus VPS4 and ATP, she saw massive constriction, as you can see from the measurements here. And again, this is about 2 times more constricted when you add IST1 and VPS4. Now, this was done in solution, and she did not see fission. And we knew from dynamine cases that when you attach tubes to a surface, they're more prone to fission because they undergo tension when you try to constrict them. And indeed, when Anna pre-adsorbed the VPS2 DID2 tubes onto the grid before treating them with IST1 and VPS4, what she observed is that in this case, they did break, and you can see the pearls that are left over onto the surface coming out of the liposome, and that really showed that the exchange of VPS2 with IST1 in the presence of VPS4 and ATP generated enough constriction to lead to fission. You may have noticed that this is actually in the wrong orientation. The ESCRT-III complex is known to predominantly work into the opposite orientation, so going inside the tube, and this is made all outside. So this is like a dynamine-like polymer. So can we get it to work into the right orientation? So I would say, yes, we can. And to do this, we actually need the full sequence to be reproduced onto NEX. So the way we did that, not me, but how Anna did that, was to use an assay initially developed by Nico in the lab, where you can endocytose silica beads with giant liposomes, forming artificial necks. And the expectation was that these necks would be very nice templates for ESCRT-III to polymerize and break them to release the beads. So indeed, Anna was able to reconstitute her full sequence onto the beads. The beads are seen by this more intense fluorescence of the membrane around them. They're very tiny, and you can see that the proteins come and bind at the neck of these vesicles, and they exchange when you put VPS4 and VPS and ATP, going first by being green and then going on with red, which is the VPS2 molecule. So we have the full sequence, and what Anna did was to test the fission of those necks with the full sequence of ESCRT-III. It turned out not to be that easy, and so she came up with a test that required pulling on the bead after it had been broken. And essentially, she used an optical tweezer to take a bead that seemed to be still attached to the membrane. She would pull on it and pull the bead into the center of the groove. And if the bead was released, when released, would diffuse inside the vesicle, that would mean that the tube had been broken. If there was a tube that would form when the bead was pulled, when the optical trap was released, the bead would go back immediately to the surface, meaning that the fission had not occurred. And then she counted the percentage of beads being broken and beads that would not break. And essentially the fission are here, and I want to point out that the only case where she had 75% of fission was the case where she had VPS2 24 D2 IST1 VPS4 and ATP put onto the SNF7 pre-polymer, meaning that she had the full sequence reconstituted on those necks, leading to the fission in the correct orientation. So what we think is happening there, and that's essentially the model, and I'm almost done with that, is that the initial transition from flat to being buckled, at least a little bit, is by addition of the layer 2 VPS224, which leads to a more rigid structure, a more rigid spiral that is more prone to buckling. And by exchanging some of the components with later components, in particular DID2, you can increase the transition to more invaginated membrane, mostly because you induce, you add another twist in the structure. And then you replace components, in particular VPS2, with IST1, allowing for constriction, and that's the fission reaction that is going on here. So now if we put this into the perspective of forming an ILV, one important component that we think is not yet addressed is the cargo. And actually, as you can see from the protein top row here, is that from this deformation point of view, we should get a membrane that is just an invagination, not a vesicle. And so we think that the cargo are there to actually form the vesicle at the tip of the invagination, and that's the neck that gets broken when IST1 is actually inducing more constriction in the DID2 polymer. So that's we think is the mechanism by which the ESCRT is functioning, we think. And actually, I'm just going to show this preliminary movie. Maybe Andela don't want me to show this, but I dare saying it's a nice movie enough so that I don't want to show you about it. We're working on that, and we think that with more simulation, there's a good chance that we understand the details of this mechanism. I think we have the big lines, but not yet the full understanding. And you will see that at the end, when you remove the last polymer, you do see fission. So besides thanking Sharon, Buzz, and Andela, I want to thank people of my group. Of course, Anna Pfitzner, that did most of the work I've shown you today. Joachim Moser von Filsek, who is the postdoc that did the cryotomography in my lab. And I'm happy to stop here and take any questions you have.
Thanks so much, Earl. This is a beautiful talk. It's great to see it all coming together, I guess decades of work. So questions. I'll give the stage to people who have not had a chance to extensively chat with you.
Can I stop sharing? Because I don't see anybody otherwise.
Okay, so let's start with Jez. He's on top of my list, and then Snezka. Jez, okay. Please.
Aurel, that was lovely. Really, really nice talk. Very cool stuff. I was really intrigued by the last stuff you showed and the combination with the first. Well, actually, I was intrigued by all of it. You presented a really nice model of VPS24 working on a SNF7 pre-existing filament to induce a deformation. And I can see that's how it would work on the context of an ILV for a multivesicular body. But for most of the other ESCRT-III biological processes, the neck is already in place. So the act of deforming a flat membrane into that bud is not required. So is the SNF7 dispensable for these forms of ESCRT-III activity? And in the last really cool bead inside the GOV experiment where you made the pre-existing necks, can you get the fission if you omit the pre-labeling of SNF7?
So it's an excellent question, of course. I would answer in essentially two ways. First, I think it's pretty clear that the SNF7 equivalent in mammalian cells is always present in all the functions that have been studied, including the ones that don't require initial flat to curved membrane transition, as you mentioned. So I think if we look at the all the functions, the first layer, which makes those flat spiral, is actually present anyway. Now we have an artificial way of looking at that by putting it on the supported bilayer, which is of course very flat, but that's never the case in the cell. And I would argue that a flat spiral would not make such a difference if you form a very large neck. For example, like in cytokinesis, where the tube would be 2 microns wide to start with, you would need actually the first layer there to accommodate a very large membrane neck at the beginning. And part of the constriction in that case would be triggered by the same transition that was seen in our experiment and Andela's simulation as a flat to invagination transition. But it could be that in other contexts it becomes a constriction transition. So you go from a very large neck to a already halfway constricted. That's my first, I would say my main thing. I think there is room still for removal or I would say bypass of some of the steps in the sequence we observe. And in particular, all the functions that are known for the ESCRT-III complexes actually have very different templates. So that means they can work on tubes that are very large or very tiny, but their dynamics is also very different. Some of them go very fast within tens of seconds, and some others will take tens of minutes to proceed, like the abscission. So I can see that you would get a full sequence in the case where you have a long time for the fission to occur, and you would have actually bypass of some of the sub-steps in the case of fast-going ESCRT-III functions.
Hello.
Lovely. I'll hand over to Snezka.
Yeah.
That was very cool. Hello. I wonder if in a real life scenario in which your bud that invaginates has tons of transmembrane domains in it, how do you think that's going to affect sort of the properties of the membrane that's, that's being invaginated? And
that's
modelable. Is it enough space actually for ESCRT to assemble? So how do you think it happens when you have this incredibly concentrated transmembrane domains in this area?
So that's a very good question. So maybe part of it is actually that the transmembrane, to create curvature, are actually not fully cylindrical. They could be asymmetric and a little bit conical, so that when the ESCRT gather them, it will actually create curvature just by forming a sort of wedge mechanism coming from these conical transmembrane domains. So actually the transmembrane, that's why it's a very good question. I think the transmembrane domain could be part of the mechanism by which the cargo actually generate the ILVs. The other proposed mechanism is actually crowding, not from the membrane domains, but from the cytosolic or the intraluminal domains, which would be on one side, and just by steric hindrance, they would just help the membrane to deform by putting pressure on one side of the membrane only. So these are the two mechanisms where I could see that clustering of transmembrane proteins by the ESCRT filament, making them densely packed, could actually trigger the deformation of the membrane.
Cool, thank you.
Thanks. Chris? And then Frank.
Yes, hi. Excellent. So I was wondering about the stoichiometry of each subunits within your in vitro experiments. Do they resemble what's in vivo, with SNF7 being much more abundant? And I ask just because, perhaps by switching subunits or filaments, by reducing the number of molecules per turn, could also play a big influence on constriction of a membrane tubule.
So the second part I didn't get exactly. So if you reduce the
from the initial SNF7 BPS32, where there might be a larger abundance of protein subunits formed within the filament.
If I remove the
tubule, switching to subunits that are less abundant within the filament.
Okay.
Per turn.
Per turn, yeah, okay. So the idea would be that the constriction is mediated because you are removing subunits that are highly abundant with subunits that are less abundant, making the overall filament shorter.
In part, yeah, with a different pitch.
With a different pitch, okay. So to answer the first question about the subunits, the only one stoichiometry within the filament I think we are sure of is when we use SNF7 alone. All the other ones, we don't really have a good idea of the stoichiometry, with the exception of the CHIM1B IST1 structure by Adam. But all the other structures so far that I know of filaments are not resolutive enough so that we can distinguish the different subunits. Of course, that's a big challenge that structural biologists want to have, because we don't know well the stoichiometry of the different subunits in the different types of filaments. And just to be clear on this, we see from the cryo-TEM structure, we see polar and equatorial filaments. We do think they are the same in terms of composition, but of course we have no way to show this. All these subunits have the similar 3D structure, and you really need to have an atomic structure to be able to distinguish VPS2 from 24 or from SNF7. So we're not yet at this stage, but it's an essential question you're hitting there, the subunit stoichiometry in the filament. So about the turnover, what we know and what is kind of known in the yeast is that SNF7 is about 2 times more abundant than any of the other subunits, which are about the same abundance, with the exception, I think, of DIT2, which is a bit lower, if I remember well, the yeast data, and I don't think it's well done in the mammalian kingdom. And again, this is from memory, so I may be wrong on those. The literature is wide on this, but what is important is that all of these seem to be tightly regulated in terms of stoichiometry in the cell. As soon as you overexpress one of them or you knock down one of them, the cell becomes very sick. Of course, you have some redundancy. So what I'm saying is to take with a grain of salt when you have, like in the mammalian, CHIM4A, B, and C. Just down tuning one will not be that dramatic, but of course, if you start playing with down tuning many of the same subunits, then you will have problems. And again, we don't really know. We know well for CHIM4B, for example, or CHIM1, but I have to admit that the latecomers did 2 and IST1. There must be redundancy in that with other components, VPS components, for example, but it has not been studied in great detail. So again, you're hitting an important point here. It seems to be highly regulated, and we don't really understand why and how this is performed by the cells.
Cool, thank you. And the last question.
Okay.
Hi, Frank.
Yeah, great talk. So just, I might have missed that, but in your model, the transition between layers, let's say when you lose layer 1 to get layer 2 must be regulated in time, right? Because that would, then the output would be different in terms of curvature and so on. So do you know what controls the timing of deassembling the SNF7 layer and?
So we don't know. That's the short answer, but we can speculate. And one of the speculation is actually that filaments that are more stretched or undergoing more elastic stress would be more prone to be disassembled by VPS4. And that brings a cool model where you could, for example, start by assembling model. You could assemble filaments on a big structure, like in the cytokinetic bridge. And you would constrict faster at the beginning because the filaments are more stretched. So there could be a link between the mechanics of the filaments and their turnover, which would be quite logical. And that's something we want to test through simulations, but also with experiments. That could explain the link we see between the time it takes for certain function to occur and the difference in terms of global geometry between these functions when you go from a nuclear pore closure to the abscission site. So again, this is area, but we think that could be a very good link to make. But it's a good, excellent question. We want to go into that direction. How you control.
Probably lipid composition might play a role, right? Also.
So the lipid composition may play a role. For the moment, what I can tell you from things we have published and things we have not published, definitely the number or the molar ratio of negatively charged lipid will strongly affect the nucleation rate and the polymerization rate of those complexes, but also cholesterol. So cholesterol seems to interfere quite dramatically with the polymerization of the proteins, and that's probably because they bind tightly with the head groups of the lipids and get inserted in between the head groups. And if you have cholesterol around, it will fill that gap and will tend to exclude the escort molecule. That's a little bit what we think. So indeed, that's a correct analysis. We do think the lipid composition has a role to play.
Thank you. Thanks.
Yeah.
Great. I think we had a great discussion. Thank you so much for the beautiful talk.
Thank you.
Hopefully you also get to visit us in person.
I will.
Next year, fingers crossed. So I think Frank said he wanted to stay and chat with you, so I think you're welcome.
Yep.
To
I can
hang
stay
out
and talk with people that want to talk.
And for everyone else, yeah, thank you for joining.
Thanks, everybody.
Bye-bye.
Yeah, great talk. I mean, yeah.